This is the sort of second part of your lab lecture this week, and generally I would recommend that you subscribe to my YouTube channel because I occasionally make YouTubes. I sent you a really nice playlist of videos to watch before this lab, and I would really recommend watching them in lieu of the lecture that we missed today. Okay, there will be a lecture tomorrow, Tuesday, at 1 o'clock if you wish to come to that, but watching this YouTube and the ones I sent earlier in the day, or I put up as a playlist, would be adequate preparation for the lab in terms of lecture. What I want to talk about is a little bit about your yeast. Okay, so over the past week, you grew some yeast. And basically what you did was you took dehydrated yeast, or baker's yeast, and you put it in some water, and the water hydrated the yeast, and you added some sugar to it, some sucrose, which was converted into glucose, and that glucose began the process of glycolysis in the yeast. And the glycolysis process, I believe, occurs in the cytosol of the yeast. And also needed for that process was a certain temperature. So you warmed up your yeast to about 30 degrees and you kept them warm for an hour at about 30 degrees. And many of you noticed bubbles coming out, okay, of your reaction. And then 24 hours later, you added some more yeast and some more warm water, I think 40-degree water, and some more sugar. And according to my colleague, Sylvia Perello, who is actually an expert on yeast, she said that probably what's happening after that 24-hour period is the yeast is running out of room to grow, and its byproducts, which are toxic, some of its byproducts, which are toxic, start to kill the yeast. So they start becoming limited by that. So by adding more water and diluting it down and then kind of re-adding the sugar and the yeast, you're kind of getting the process going again, okay? And what we were trying to do was to use an enzymatic reaction to do a traditional organic chemistry reaction. In other words, we're going to hijack an enzyme that's in the yeast, in the ribosome of the yeast, to do this chemistry, to do some real organic chemistry. And this was very popular in the 90s because it was viewed as a way to make molecules in one enantiomeric form or in very high enantiomeric excess. And this is because enzymes are chiral and they usually have a pocket in them where the chemistry takes place. And that pocket is very asymmetric, and frequently it only allows the chemistry to occur from one face. And the enzyme that we're going to talk about is no exception. Okay, so that's kind of what you were doing. Sylvia's probably a good person to talk to about yeast because she does all her research on yeast. So not yeast, but on yeast, on an organism, a yeast organism. Okay, so the general process that I'm talking about here, that we were kind of hijacking, right, is that you start with glucose, right? And I'm writing this very generally, okay, right now. We're going to study this enzyme again at the end of the year, and you'll learn more about it. But glucose plus 2-ADP, okay, plus 2 inorganic phosphates will yield ultimately, okay, after glycolysis and then fermentation or anaerobic breakdown of pyruvate, will yield ultimately 2 molecules of ethanol, an organic molecule, they're all organic molecules, plus 2 molecules of CO2, and then of course 2 ATP. That's kind of the net process. You're going to say, what is a net process? That's a net process. But realize in this is glycolysis, which results in pyruvate, okay? And then the pyruvate is broken down further, okay? So glycolysis, which would be a lecture in itself, I suppose, produces pyruvate, okay, which is pyruvic acid. And if I were going to draw the structure of that, because I am an organic chemist, I love to draw structures. So in biological systems, it would exist in this form, and you actually get 2 equivalents of pyruvate, because if you do the mass balance, Glucose has 6 carbons, so this would account for the 6 carbons of glucose. And then yeast does sort of a curious thing, okay? Though the glycolysis is an aerobic process which occurs in the cytosol, what yeast does is yeast takes pyruvate, and rather than going through oxidative phosphorylation as it would in your body, okay, normally, to produce CO2 and water, it is anaerobically converted into acetaldehyde, okay, and CO2. Okay, so if we did the mass balance on this, you'd get 2 of these and 2 of these. Okay? Then what happens is this molecule, which is acetaldehyde, is converted into ethanol. And of course, this is how you brew beer, okay? So you get 2 equivalents of ethanol. So that's how it ends up. And you might say, well, why? Why does it do this, okay? This process, the process of taking the acetaldehyde and converting it into the alcohol, is very important to yeast because what it does, so the process of taking acetaldehyde and going to form alcohol and CO2, or the alcohol, requires a molecule of NADH. So for every acetaldehyde you need 1 NADH molecule, and I'm going to show you that structure in a minute. And what this does is this regenerates NAD+. And NAD+ is needed for glycolysis. So really, without this process, without this happening, you would not produce the NAD+ that's needed for glycolysis, and the glycolysis is where most of the energy comes from for the cell. Okay? So I've tried to explain that a little bit. We're going to be focused just on this one little reaction. We're going to hijack this reaction. Now, what is the enzyme that catalyzes this process? The enzyme is called yeast. There's actually several of these, but yeast alcohol dehydrogenase. And yeast actually has more than one yeast alcohol dehydrogenase, but we're not going to get into that. We're going to kind of focus on the main one. Now in humans, okay, you have many alcohol dehydrogenase, several, I shouldn't say many, but you have a number of alcohol dehydrogenases, and they're generally used to convert alcohols into various aldehydes and ketones that you need to build metabolites, to build molecules that you need. So the reverse process is actually more important in the human body. However, if someone drinks too much, if they drink too much alcohol, that what your body does is it takes the alcohol in the human body and converts it back into aldehyde, okay? And you know, it's kind of interesting. I was reading, I don't know, it might have been on Wikipedia, but I was reading that it's kind of an interesting thing, you know, that young women apparently do not process alcohol as well as young men, and then middle-aged women process alcohol better than middle-aged men. And it's something that varies genetically, okay? So it kind of accounts for the fact that some people can't handle alcohol or can't process alcohol. At least that's part of it. But we're going to again, in yeast, this is the major process, although if enough alcohol built up, there's another enzyme that would bring the alcohol back again. So it's very interesting that yeast can kind of produce alcohol, ethanol, and can actually chew it up again. Okay, but again, that kind of attests to the fact that, you know, biological systems are feedback systems, and biological systems, reactions in biological systems are equilibrium. Okay, so that's kind of like the general idea. Now, what are we going to do? We're going to hijack this system, okay? So instead of, what we did was, we had the acetaldehyde in there, of course, because it was made from glycolysis, from the destruction of the sugar, or from the metabolism of the sugar. But what we put in instead was this molecule, ethylacetoacetate. Okay, this is just a regular old organic molecule, and you threw it into the reaction after you got the yeast hydrated and after you got the yeast producing CO2. So you got this process going, and then when you got the process going, you threw this molecule in, and your hope was that the molecule would bind into the binding site of the enzyme. Okay, now unfortunately, I usually have a nice movie to go along with this. I used to have this very nice movie of how it works, but roughly what I'm going to say is their yeast alcohol dehydrogenase consists of 2 subunits. Each subunit has 2 zincs in it, positively charged zinc. There's kind of like a binding site in proximity to the zinc. One of the zincs is in the binding site. The other zinc is just structural and kind of holds the subunit together. The 2 subunits are the same and they do the same thing. So each subunit can kind of process one molecule at a time. Basically what happens is, there's a pocket, there's a zinc, okay? The NAD, which we're going to talk about in a second, the NADH is very, very complicated structurally, and I'm going to show it to you in a minute, and it's chiral. It fits in the pocket in a very specific way, and then the aldehyde, or in this case, the hijack molecule, fits in a very specific way, and then the NADH delivers the hydride to that carbon. This group opens up, and it picks up a proton. All right, that's being very simplistic, but there's a pocket. It's got this very irregular shape. That shape, the NADH just kind of slides in and has a particular position in the pocket, binding to the zinc. The aldehyde molecule comes in, the oxygen binds to the zinc. They line up with specific faces facing each other, and then this delivers a hydride to that. Okay, so that's what should happen. That's the biological reaction. However, we're throwing this in, all right? And the question is, will this behave like this? And you might want to think, well, how is this a mimic of this molecule? You really want to look at this end of it, because this is the part of it we were trying to reduce. So we are hoping that this part of the molecule will behave exactly like acetaldehyde, and the NADH will deliver the hydride to a specific phase. Okay, so let me show you how an organic chemist would do this reaction. Okay. So an organic chemist, and some of you have had this already, an organic chemist would take a molecule, it's like say they had this aldehyde, and one way you could do this reaction is you could add LAH. So I think the students from the fall who had Dr. Schmink, you've probably already seen this reaction. My students are going to see it really soon. Okay, it's coming up very, very, very soon. The idea behind this reaction, it's a reduction. It's the opposite of an oxidation, right? A reduction, an oxidation is the loss of hydrogen, right? Or the gain of oxygen, nitrogen, or halogen. Am I getting in some trouble here? Okay, I'm not talking about real trouble, students. I'm talking about time. Reduction is the gain of a hydride or the loss of oxygen, nitrogen, or halogen. We talked about that in the fall. Okay, what's special about lithium aluminum hydride, and I'm using it because it's really brutal and it kind of really illustrates how uncontrolled LAH reductions can be. What's special about this is even though there's a negative charge on that aluminum, the aluminum is a metalloid, if you look at the periodic table, and the hydrogen here is really more negative than the aluminum. So the hydrogen has a delta minus. And so what this reagent does, I'm writing this very crudely, right? If you put your deltas in here, this would be delta plus, and this would be delta minus because of the oxygen pulling electron density away in the sigma bond. The hydride would come in, hit this carbon, open this up, and I am vastly oversimplifying this, okay, for the point of getting into the biology. Okay, you would get this. This is initially complexed onto the aluminum. I don't know. I know Professor Bruce, our textbook, tends to oversimplify these a bit. Just makes them very general. But this is complex to the aluminum. And then in a second step, what you would do is you would, and I am oversimplifying it, okay? So I openly admit that. This gets protonated. It's more involved than that. And then you would get the reduced form of the molecule. So an alcohol, this is the CH3 group, an alcohol is a reduced form of the molecule. Okay, so the reduction is this part when the hydride comes in. So the question is, how does this happen in biology? And as I said, it happens by the molecule fitting into that little pocket. Okay, and NADH is the reducing agent. Okay, here LAH is the reducing agent. So in biology, there's more than one reducing agent, but one reducing agent is NADH. So biologically, what happens? Now, you're going to have to put up with me drawing this molecule for a minute. And again, I'm an organic chemist. And because I'm an organic chemist, so essentially, I'm probably being a little, I'm just going to put it like this. Again, this is complex to a zinc, okay? But also complex to this is the NADH. Now, what is NADH? You're going to be sort of horrified by this, and I hope I can draw it small enough. But this is the important part of the NADH. I'm going to show you this, and it's very interesting because, of course, can you get that on the video? Mm-hmm. Of course, it's an organic molecule. And what I love about aromatics, I've been saying this in my lecture class, aromatics are like, they're like electron sinks, or they're like wires that electrons can transfer through. And you see this frequently in biology. So this is hooked into a ribose. And organic chemists care about structure. We really care about it, so we always draw all our structures out very carefully. Hopefully I'll draw this one successfully. Okay, this is called nicotinamide adenine dinucleotide hydride. It's in its hydride form. And I don't have it drawn in its hydride form here, so hold on a sec. I have it drawn in its oxidized form. Okay, there's a lone pair here, so let's keep track of this. This is really going to tie in with what we're doing in lecture, the lecture part of the course. So then there are two phosphates. I hope this is readable. I'm kind of worried about that. So far, so good. Mm-hmm. Okay. Okay, those are the two phosphors, nicotinamide adenine dinucleotide. Okay, and these are the dinucleotides. Then this is linked into another sugar, another ribose unit. And the riboses are involved in the protonation and deprotonation, but we won't get into that right now. And then this is hooked to another nitrogen base. I hope this is readable. I will do it again in lecture, and I'll try to tape it tomorrow in lecture. Now, the reason I got into this in such detail is, again, I'm an organic chemist, but also I want you to recognize these nitrogen bases, okay, from the compounds that I made you learn. I said to you, you needed to learn some of the aromatic structures because they were part of biological molecules, okay? So in my lecture, I've been talking about these. So you can see here, this is a relative of pyridine, right? And this is a relative of pyrimidine, okay? I'm sorry, purine. That's a purine derivative. Okay, this also has a carbonyl here and an NH2. So I apologize for that taking so long to write. Now again, what's all this, Well, again, this has this very complex structure kind of like wraps around and sits in the pocket of this enzyme subunit. And then what happens is the hydride from this attacks this. So where does that hydride really come from? Well, what I want you to think of is the driving force, maybe part of the driving force, if you think of it like an organic chemist, is aromatization. And I apologize, I'm writing on my terrible board here in my house. All right, so think about this. If I kick these electrons in, this is like an electron drain, and these electrons come up. This will come off as a hydride. And again, I'm saying it's really sitting in the pocket. I just have this drawn out for convenience. This will hit this will open up, and ultimately, this is going to pick up a proton. And there's a whole complex proton transfer that occurs. So this NADH is this, okay? And then look at what's happening there. When these electrons come in and these go up and that comes up, that becomes an aromatic ring. And realize too, why is this a catalyst, right? A catalyst is creating a place for the reaction to take place, okay? Like a little pocket for it to take place. The molecules get aligned properly. Our reactions, we heat them, the molecules are colliding, they're knocking into each other. It's very chaotic. In enzymes, everything's very ordered, okay? So the probability of this happening goes way up, okay? The energy goes way down. It can happen at physiological temperature, or the yeast temperature, which is 30 degrees. And it happens very specifically. In other words, the hydride only comes in on one face of that, even though we're not making a chiral molecule, okay? So then what happens when this happens? This will go into the form where it is the alcohol. And again, you're saying, I don't care that that's happening because that's not chiral. But we did this reaction with a different molecule. We hijacked the system, okay? So sticking it instead, what did we do? Instead of this molecule, we reduced this molecule, which, again, presumably it's fitting in that pocket somehow, okay, of the enzyme, okay? So the hydride's coming here and opening that up, okay? Now I want you to think about this. So then what's the outcome? The outcome is you get only, I think it's like 94 or 97%, I shouldn't say only, but that's tremendous enantiomeric excess. I believe the S compound. So let me see if I can draw the S compound. That should be the S compound. You get mostly that, which is saying the hydride's coming in from one side. And then the other things I want you to really note are it's not only stereo selective. Notice I'm saying selective, not specific, because I'm getting some of the enantiomer, but it's also regio, or maybe better said, chemo selective. Okay, chemoselective. I know that's how Dr. Mal would say it. Okay, so what do I mean by chemoselective? Well, if I took LAH and hit this thing, LAH would blast this into an alcohol and would blast that all the way down to an alcohol. It's not very chemoselective. However, the enzyme is very gentle, it's very chemoselective, and it really selects for the ketone over the ester group, which is really an amazing thing. Okay, so am I okay for time?
It's almost 24 minutes.
All right, perfect. Okay, so that's kind of like the theory, and I think that's pretty good as long as you can see that. Okay. What I want to do is a quick flow chart. I just want to give you a quick flow chart of this so you know what you're doing and what you're not doing. Okay, and that'll only take about 5 minutes. So this is exactly how long I want it to be. Okay. Now, what are you going to do? All right, you've already done part of the experiment. You've made your yeast. So what I suggest is you go into the lab. You start out with your Diels-Alder. Take a look at your Diels-Alder. I gave you some scenarios to deal with. We're going to figure out how we're going to approach your Diels-Alder. But then, if your Diels-Alder isn't crystallized or you've recrystallized it and sitting and crystallizing, leave it alone. Don't fuss over it. Chemistry goes better without you fussing over it. As my mother used to say, this is like probably a terrible saying, but children and plants do better with less attention. And I would say the same thing about certain parts of chemistry, particularly recrystallization. People who fuss over their recrystallizations invariably boil off all the solvent, or they don't put the right solvent combination in, and they just get too impatient. It takes a long time to grow crystals. Sometimes I grow crystals over weeks, not over 5 minutes. So please be patient. Okay, so while that's doing its thing, ignore it. What you're going to have to do is add, so I'm going to make a little flow chart. This is not a bad way to put things into your notebook. Add zeolite to your goopy yeast mixture. Okay, get it off the stir, add the zeolite to it. It's a certain quantity. You have to have scaled this down. Zeolite is diatomaceous earth. Diatomaceous earth is literally like little organisms, like little microscopic organisms, shell that have sort of a hard surface, and it acts as a filtering agent. So when you're trying to filter these yeast cells out, they get all caught up in the diatomaceous earth, and that helps you get rid of it. It's really kind of a nasty filtration, I must tell you. Now what you're going to do is vacuum filter through a sintered glass funnel. And for some of you, this may be a new experience. Okay? A sintered glass funnel is a funnel that has built into it sort of like a frit. And the frit's made out of glass, and the glass has pores in it. Okay? And they are of a certain diameter. Now, frankly, we don't have the most amazing set of sintered glass funnels. Some are a little more porous and some are a little less porous. We don't have the exact porosity that's required in that experiment, but I thought it would be a good experience for you to use it. The advantage of this is that rather than putting cellulose filter paper in that might absorb some of your compound and some of the cells and the biological material, the material is just going through glass. So there's no interaction. So it's like little pieces of glass that have pores in it, and it's built right into the funnel. They're very fragile. And it's done like a normal vacuum filtration. Now, what I recommend in a group is divide the solution in 3 or in 2 and use 2 different funnels or 3 different funnels. You can have more than one setup because you've got this big group. You could have one person working on the Diels-Alder and the other 2 people filtering. Okay? The filtration takes a while. If the filtration gets really nasty, you might consider at the end using our centrifuge. So in other words, again, what are you trying to do here? You're trying to filter out the yeast, okay? And all the particulate matter that's in the mixture. Am I okay? Mm-hmm. So, at the end, sometimes it gets so gloppy and so sticky that you can't get any more through. It's just impossible. You could take the remaining liquid, divide it between 2 centrifuge tubes, and centrifuge it down. And the TAs and myself will be happy to show you how to do that. I don't think everyone can do this from the get-go because we only have 1 centrifuge. So I think everyone has to do some filtering. Okay, once you get it filtered, you're going to have 2 components. Of course, you will have the filtrate. Oh my goodness. And you will have the residue. The residue is what you want. Sometimes people don't realize, I'm sorry, the residue is what you don't want. The filtrate is what you want. The filtrate, the organic molecules are going to go through. So in other words, if you actually reduce that ethylacetoacetate, any unreacted ethylacetoacetate and any of the resultant alcohol are going to go through with all the aqueous solution and sucrose and sugars and things like that are still in there. So the filtrate comes through. What you want to do with the filtrate is you want to extract it 5 times with ether. Okay, you have to be careful with ether, all right? Very fumey, you got to work in the hood, and you've got to get used to the idea that ether's density is different than dichloromethane. So you're going to have to figure out which layer is which and try to review all that, okay? I'm not going to write this over and over and over, but every time you do this, you will have an organic layer, which is the ether layer, and you will have an aqueous layer, which is really the filtrate again, okay? And what you're trying to do with multiple extractions is to get all of our organic molecules out, and it's amazingly selective. except for getting some ethanol out, if you have ethanol in there, you really just get starting material and product out of this. So it works pretty well. The only thing I'm worried about is these were not the most bubbly reactions I ever saw, and I did buy new yeast, but I'm a little worried about my yeast. Okay, the organics all get combined. Then what you're going to do is dry over mag sulfate, which you've done before. I want to do a traditional dry where you just add the mag sulfate till it stops clumping. Remember that? That's always like a painful process. Then you're going to have to gravity filter. Okay? And then after you gravity filter, okay, you'll have this ether solution. And again, keep yourself in the hood and keep things covered up. Okay? So you'll have, after you gravity filter, you will have filtrate. Now, in this case, the filtrate is ether in nature with your organic molecules. You'll have the mag sulfate as the residue. We're going to dispose of that. If you want to rinse this with a little ether, you can. But then this filtrate, what you want to do is rotovap to dryness. This is where we have to be careful, okay? We are not going to distill this stuff. I don't consider it safe to distill anything that had ether in it, okay? So you're going to rotovap to dryness. Now, we have a lot of rotovaps now, which is great. But we have found that when you use the dry ice rotovap, you can get almost every volatile molecule off this thing, okay? So even if you're put on a regular rotovap first, I would put your sample on the dry ice rotovap for a few minutes to get it down to its essence, all right? And then what are you going to do with this? You have to get a mass, okay? You have to get a NMR, and you have to get an IR, okay? The IR is going to be a liquid between salt plates. So it's the Oreo cookie. It's not the film. It's like you're going to take a plate, put one drop of your sample, put another plate on, and run the IR. And I think you're good at running IRs. Now, NMR is always a problem in these experiments, these couple weeks where we do synthetic stuff. It is groups. We did miss a day this week, so we're going to be backlogged. It's possible you may not get on the NMR on your day. Next week's lab is the fluidamide synthesis. It's relatively simple. I'm envisioning some people finishing this, or maybe doing the last bits of this next week. So just be mentally prepared for that, okay? All right, so that's it. So I'll see you in lab.